Welcome back to the course Fundamentals of General Immunology. So today we'll start our module 5, and the title for that module is Immune Signal Transduction and Activation Thresholds. So till now we have understood how B and T cells are selected, how these receptor editing could be very specific towards the cells which are self-reacting in nature, reacting to the self-antigens, and therefore these are getting eliminated. And further, we also looked into some of the basic concept of tolerance. In that, we have discussed about the central tolerance, peripheral tolerance, and to some extent, we also discussed about the feto-maternal tolerance, which are happening in case of females post-pregnancy. So today, we'll discuss further details about receptor signaling cascade in B and T lymphocytes. So essentially, we have understood how B and T cells are selected, and now we will try to understand how these receptor signalings are individually working with B and T cell population. So we'll go one by one. So first we'll start with the B cell, and then later we'll look into the T cell in general. So when we discuss about the B lymphocyte selection, we discussed that say we have a new receptor specificity which are expressed. So essentially we have this immature B cell, and these are the antibody which are expressed on the surface. If the new receptor is self-reacting, the B cell undergoes apoptosis that we have discussed in a detail. And if the new receptor is no longer self-reactive, the immature B cell migrates to the periphery and then mature as a functional B cell in order to do the function. Now all these phenomena are controlled very critically by receptor mediated signaling pathway. So since these are the receptor B cell which are present on the B cell, then there are other receptors which are present on B cells. For example, there are CD specific such as CD19 which are present on the B cell, and then there are other proteins which are present. How these proteins are getting activated upon binding to the ligands, and how these receptor signaling can happen in detail, we will try to understand in this lecture. So before going to that details about the receptor signaling, we need to understand few concepts, and the very critical first concept is phosphorylation. So what phosphorylation stands for, that we have always discussed that a protein is getting phosphorylated. Phosphorylated is a post-translational modification. So what is happening when a protein is synthesized in the cytoplasm after ribosome is doing all its function, then protein goes to the different organelle or any kind of other cellular compartment to do modification in order to do its proper function. One such modification is, suppose we have a protein, the protein is getting a phosphate group from adenosine triphosphate, and then the adenosine triphosphate can transfer one phosphorus molecule and then convert itself into adenosine diphosphate. So let's try to understand it in a detail. So suppose this is a protein which you can see here, and this protein might have multiple amino acids that are responsible for phosphorylation, such as serine and threonine. Tyrosine also one such molecule. So serine and tyrosine are specifically the target for phosphorylation. So what's happening that once there is phosphorylation event, one of the protein which comes into picture is called kinases. Now kinases are a specific protein which can allow the addition of phosphate group into a protein. So because of phosphorylation, the kinase can convert adenosine triphosphate, which is here, will convert it into adenosine diphosphate. That means there are two phosphate group here, and one of the phosphate group will be added into the protein. And this protein is now become activated. And because of this activation, this protein can do a lot of functions, which we will study in the receptor signaling cascade later on this lecture. Now, once the function of this protein is over, this protein is no longer required to do some event, then this phosphate need to be removed in order to convert the same protein into the native form. The second enzyme does play a very important role here, which are called phosphatase. Now phosphatase can remove the phosphate group and can convert the activated form of the protein into the native form of the protein by a process called dephosphorylation. So the phosphorylation event is happen because of the presence of an enzyme called kinase, and dephosphorylation can happen because of the presence of phosphatase as an enzyme. So the phosphate can recycle into the system and do the function individually. So this is the one very important post-translational modification that can happen in the protein, particularly the protein which are involved in signaling cascades. So phosphorylation does play a very important role. Let's try to understand another typical the event of kinase when it comes to the surface protein. So we'll try to see the receptor signaling and phosphorylation cascade. So we understood that the kinase can do phosphorylation and phosphatase can remove the phosphate group. Now let's try to see. So there are multiple proteins which are present on the cell membrane. So this is cell membrane here, and these are the proteins which are present on the cell membrane spanning this is a extracellular domain, this is an intracellular domain, and this is a transmembrane domain. So the protein could be divided into three parts, where the outer surface, which is called extracellular domain, can bind to the ligands here. So any of the ligands such as an antigen or any kind of protein, nutrients, any signaling molecule can bind to this protein on the extracellular domain, and then there are a signaling cascade which can be activated downstream, which are in the cytoplasmic tail. That means this is cytoplasm. So something present in the cytoplasm can initiate the cascade downstream. So once there is a binding, then we can see that this particular domain gets activated, and because of this activation, the inactivated protein kinase become activated. And so there is an activation of the protein kinase, and once the protein kinase is activated, then we can see that one of the protein can be converted into other form of the protein by addition of the phosphate. So we can see that activated protein kinase can act as an enzyme which can convert one of the protein into an active form by adding the phosphate group into it, which may be possible that there could be other downstream protein which require the phosphorylation of this protein. So then again the other inactivated protein can be phosphorylated similarly with the help of the activated kinase. That means there are multiple stages of the phosphorylation that can happen when there is an addition of the ligand on the surface of a protein which are exposed outside. This could be a B cell, this could be a T cell, or could be any other cell type. Since we are talking about immunology, we'll concentrate our discussion upon the B cell and T cell activation pathways. So what ideally this slide tells us is we have a protein. Once the protein is bound to another protein which is coming from outside, it can activate a kinase, and the kinase can do phosphorylation, and this phosphorylation can activate multiple protein inside, and this activated protein can do function. So the event of phosphorylation which we discussed in the earlier slide was actually visible here when we see that the proteins are getting functional at different stages here. So for example, here we have seen that there could be a different kind of proteins involved in a stepwise manner. So first is one activation, second is another activation. There could be possibility that third is activated, and essentially without that activation we can't see the functionality of the cell. That is what we learned, how the signal is getting transmitted from one protein to other protein so that the cells become functional. So this is one event of phosphorylation, how it can cascade there. Now since we are talking about the protein which are getting phosphorylated with the help of a kinase, there could be possibility that the receptor itself is having a kinase domain or kinase function, and those all receptors are called receptor tyrosine kinases, RTKs. So these are again very specialized receptor which are inbuilt having a capacity to do a phosphorylation event, autophosphorylation event we used to call. That means they have a capacity to self-phosphorylate themselves. So for example, here we have signal molecule which are coming from outside, And generally, we can see that there is an event of dimerization. So two of the signal can dimerize, and then essentially we have receptor tyrosine kinase. Why there is a tyrosine involved here? Because tyrosine is a particular amino acid which actually do phosphorylation, which actually phosphorylated in order to do the function of a protein. So here it's a domain called tyrosine kinase domain. So these two proteins which are there in the membrane, after binding to this dimer protein, is getting dimerized here. And once there is a dimer formation, we can see there is an stimulation. And this stimulation of receptor tyrosine kinase can do an autophosphorylation. So we can see that there are addition of the phosphate group into this receptor tyrosine kinase. And this phosphorylation of receptor tyrosine kinase can recruit other proteins into the site, which actually require to do functional aspect of these receptor tyrosine kinases. So what was the basic difference between the earlier slide and this slide is we have a phosphorylation event happening by recruitment of a kinase, because kinase will come and do a phosphorylation by utilizing the ATP. These are all active processes because the phosphate is coming from ATP and there is involvement of energy. So this is actually an active process altogether. In this case, we are trying to look into how the receptor itself is acting as a kinase. So where the receptor can bind to the protein or the ligand or the signal molecule which are coming from outside, and here we are trying to see that there could be a possibility that two protein get dimerized, and the dimer form can bind to the receptor tyrosine kinase to make themselves as a dimer. And further, this can activate their autophosphorylation, and autophosphorylation can next time recruit multiple proteins into their site so that the cell become do its proper function. Again, emphasizing the same point, this could be a bit different cell type altogether. So after understanding the tyrosine, importance of tyrosine because it is getting phosphorylated to activate the site, the phosphorylation event, the kinases, and the event of downstream multiple step phosphorylation in order to activate, let's try to understand the B cell receptor. So why we are trying to think about this activation pathway in context of B cell, that is what we'll try to see in the next slide. So let's see, this is typically a B cell receptor. So what's happening that essentially B cell receptor is nothing but the immunoglobulins, and we have looked into the immunoglobulins that there are 5 different kinds of immunoglobulins. Let's try to recapitulate one by one. So there is M, A, D, E and G. So there are 5 distinct kind of immunoglobulins which are there. IgG is the most abundant one, and the IgM is the most, say, having pentameric structure, so that can bind to a multiple antigens altogether. Now these is actually called B cell receptors. So these are the variable domain we can say, and these are the constant region, and these are the heavy chain, and these are the light chains. So we have gone in details about it, so we will not go and discuss about these protein again, but try to get an idea that these are present on B cell. So this is actually a B cell plasma membrane. So these are the transmembrane protein, and these are actually a transmembrane domain. Apart from BCR, there are many other proteins which we are going to discuss. One such protein is called Ig alpha and beta. So these are again the dimeric protein, and both are coming in close contact with a disulfide linkage. We can see here it's a disulfide linkage, and they have a long tail in the cytoplasmic area. So we can see that these are the small extension of the protein into the cytoplasmic area, right? So we are having an extracellular space and cytoplasmic area, and this extracellular space contains a very specific kind of a domain. These are called immunoreceptor tyrosine based activation motifs, or ITAM. So immunoreceptor tyrosine based activation motifs are basically tyrosine rich molecules which are actually phosphorylated in order to do signaling cascade downstream. So there are other protein, as I told, there are CD19 also present here, and there are other proteins also which will be there in the surface of B cell, but these two are critically important to do a function of B cell in general. So let's move on and see some more details about the signaling cascade. So when we discussed about that the binding of an antigen or a receptor from binding of an antigen or a substrate from outside can start the cascade process. So the first thing is the antigen induced cross-linking. The process is the very first step of the activation or the receptor signaling cascade is the binding. So the complementarity determining region, the CDR of a immunoglobulin or a B cell receptor can bind to antigen, and there will be a cross-linking. So two of the immunoglobulins will come together, and this could lead to the phosphorylation event to happen, and phosphorylation can be done by kinases. Right? We have discussed that these are the protein. Now there are many kinases present in the body, and we are typically trying to understand the kinases which are expressed in B cell. And in the B cell we have a very specific kinase called SRC family kinases. So these are actually kinases which are involved in activation of B cell. So SRC family kinases, some example which are listed here, and we'll try to look into in details in the next slide, is such as LYN, LYN, FYN and BLK. These are some examples. There are 9 total SRC family kinases which are reported till date. Will do a phosphorylation of item molecule which are present in the Ig alpha and beta of B cell receptor. So I also wanted to emphasize that these are also called as, say, we'll go in the next earlier slide and see that these are also called as CD79A and B. So we can also say that these Ig alpha and beta chains are also called as CD79, can also involve in the signaling cascade, but for our understanding we'll keep the discussion as Ig alpha and beta. So what we have understood is once there is a binding, then there could be an activation of SRC family kinases which can do a phosphorylation, and this phosphorylation can happen very specifically on this ITAM domain which are present in the cytoplasmic region of the protein. Now what next? So the protein will essentially involve in a variety of process in order to do an activation. So we need to understand how these kinases behave. So let's try to understand this SRC family kinases. As I told earlier, so there are 9 members which are reported till date, and these are named by 3 letters such as SRC, YES, FYN, FGR, LCK, HCK, BLK, YRK and LYN, LYN. So there are many other which are involved in the B cell activation or any other cascade related to that, but these are some of the 9 SRC kinases which are reported to be involved actively in the activation of B cell and other T cell population. So these functions of SRC family kinases is multifaceted. They can be involved in cell growth, their adhesion, inflammation, which we have discussed earlier. They can help in the cellular survival, and particularly in the central nervous system, the brain and the spinal cord, they are involved in neuroplasticity. That means the involvement of one cell type, development into other cell type, and there could be a plasticity which we have discussed, that the neuronal cell can be converted into some glial cell or some kind of an astrocyte which are doing a multifunctional role. But the proteins are very dynamic in terms of its activity. So when we see this protein SRC family kinase very closely, let's try to understand it, how this an inactive protein kinase can be converted into an active protein kinase. This is just for your reference because this will help you understand how this protein modulation can happen. So we can see that this protein is having an N-terminal domain and the C-terminal domain, and then we have multiple motifs which are or domain which are present inside the protein. So this is called as SH2, SH3 domain, N lobe and C lobe, which is actually a kinase lobe. and there could be a tyrosine molecule which are present in 527 position of this protein, which is actually phosphorylated. So this is an inactive SRC kinase. That means an inactive SRC kinase will have a phosphorylation of tyrosine at 527 position of that protein. Again, there is a point that intramolecular SS3 binding to protein-rich region of the linker segment can cause the inactivation of SRC. Again, this is just a reference point to remember that there could be multiple events that might be happening, but one such thing which is pretty straightforward, that there could be a phosphorylation event of tyrosine at one particular location. Now this is inactive. What will happen when this become phosphorylated and become more activity wise? So what will happen when it gets activated? So the activation start with the phosphorylation of SRC activators. So essentially what will happen, the SRC activated molecule, which is actually bind to the protein rich region of the SS3 domain, which was discussed earlier, and then essentially here the 527 position is getting dephosphorylated. So the inactive form is phosphorylated and the in dephosphorylated form is become activated, and further phosphorylation of another molecule at 416 position actually activate this protein. So making this story short, what it implies that the protein which does phosphorylation, so these are actually kinases, these protein in turn also controlled their functionality by phosphorylation. That means there are multiple steps of phosphorylation and dephosphorylation that can make the protein active, because these are kinases. They can in turn do kinase activity to other proteins. So these are all complex proteins. These are controlled very critically in the cellular level. Many functions of these proteins are explored by scientists, but many of the functions of these proteins are still yet to be explored. So why we are discussing these SRC family kinases, because these are critically controlled and expressed in the B cell population. We try to see that what is happening next. So what we discussed, that the antigen induced cross-linking leads to activation of SRC family kinases. We have looked into what is SRC family kinases, and these SRC family kinases can do phosphorylation of the item molecules. Now what will happen next is this phosphorylation event of the item molecule can recruit multiple other proteins as well into the cytoplasm area. And these are some of the proteins which are listed here, such as phospholipase C kinase, SLP65, this is another kinase, BTK, this is another kinase, and GRB2, that's another kinases. So there are many different kind of kinases which are involved in a stepwise manner. So there is a phosphorylation of phospholipase C, then there is a phosphorylation of SLP65, which can dock the another phosphokinases called BTK, which again do a phosphorylation of another phosphorylating protein called GRB2. We'll see it just in a moment, but what I'm trying to emphasize here, that the phosphorylation event, one receptor phosphorylation can do an event which can further lead to multiple other phosphorylation that can lead to activation of the cell protein. So this one such thing which I forgot to mention here is docking of the SYK. So this is actually a spleen based tyrosine kinase. S stands for spleen, Y is actually a notation for the amino acid tyrosine, K is a kinase. SYK is a kinase which are actually spleen tyrosine kinase that are getting docked into the site post item phosphorylation. So once there is a phosphorylation of item molecule by SRC kinase, then SYK protein will come and SYK will do a phosphorylation cascade further. So let's try to look into these what are these important kinases which we have listed here. So BTK is actually a Bruton tyrosine kinase. SLP65 is actually also known as Blink, BASH or BCA. GRB2 is a growth factor receptor protein bound protein 2. which is again a very critical protein involved in cell growth. So actually this SOS is another protein which is called son of 7-less. This is the protein again involved in multiple growth, promotion and related to cellular proliferation. So GRB2 is an adapted protein that acts as a crucial molecular bridge connecting activated receptor tyrosine kinases to the son of 7-less, which is SOS protein. So essentially these are all very specific proteins which are involved in B cell activation cascade. So that is what we need to understand, that these proteins does play a very important role in activation of the B cell. So till now what we understood is there is a binding of antigen, and binding of antigen can activate the SRC family kinases which can phosphorylate the item. Item phosphorylation can recruit the spleen tyrosine kinases, and the spleen tyrosine kinases can do phosphorylation of phospholipase C kinases, SLP65 kinases, BTK, GRB2 and SOS. And further, these all activation can lead to the B cell downstream signaling cascade. And what is that is these can activate some of the important proteins such as nuclear factor of activated T cell, NF80, nuclear factor of kappa beta or kappa B, a nuclear factor of KB cell, and there is another protein called AP1 which we will try to look into a little later. And these are all transcription factor, and when we are talking about a transcription factor, their location is always inside the nucleus. So these proteins are getting activated upon this cascade, and these are directly moving inside to the nucleus, and once they are moving inside the nucleus, they can bind. They are a transcription factor, so they can bind to a specific DNA molecules and can start transcription of the gene, which could lead to further production of different kind of cytokines or other kind of molecules which can helping the B cell to do the function. That means these B cell receptor kinases or the B cell receptor biology is essentially ultimately activating lot of transcription factor which can start transcription of the multiple genes, which can lead to the production of, which could lead to a different cytokine production in the cellular mass. So what we discuss, AP is actually called activated protein 1. It's actually a dimeric transcription factor complex composed of protein from, these are again a very critical name, such as Jun, Phos, ATF and MAF family, containing a very specific leucine jeeper. So there are stretch of leucine amino acid present in this protein which can specifically bind to the DNA area to do transcription. So this is all about the B cell receptor signaling cascade, which is critically controlled by the phosphorylation event or the kinases recruitment. So we will try to move on and try to understand the other term, which is called the B cell signaling. Now in B cell signaling, we have discussed this event. So let's try to see the event of B cell in presence of a complement and upon the virus infection. So for example, here we can see that the complement protein can be activated when there is a microbe coming into the picture. So once there is a entry of a bacteria or any microbes, the complement protein can bind to those microbes, and this complement protein binding can activate another set of reaction. So there is a complement protein called C3d, and this can bind to the microbes very specifically, and once there is a binding, this can attach themselves to the CDR region of immunoglobulins, and here we are trying to understand IgM molecule. Apart from that, we have something called complementary receptor 2. So the complementary receptor 2 is also present in the plasma membrane of B cell. And apart from that, we have CD19 and CD81. So we need to understand that apart from BCR, B cell receptor, which is immunoglobulin, Ig alpha and Ig beta, there are CD19, CD81 and CR2. These are the three other proteins which are present on the cell surface of B cell. So these are also considered as part of B cell receptor, although we are not calling it a B cell receptor, but they are involved in multiple processes. So once there is a binding or complement to the microbes, the complement protein itself can bind to the complement receptor present on the B cell, and once there is a binding, we can see that again the SRC family kinases can be recruited here. They can do phosphorylation of the item which are present on the cytoplasmic domain of the Ig alpha and beta, and this can recruit the spleen tyrosine kinases which can again do a phosphorylation and ultimately leading to the kinases activation, which can further downstream activate multiple other proteins, leading to activation of B cell, which can do further killing of the pathogen. So essentially this activation is not possible unless until we can see this activation cascade. So in order to neutralize these microbes, this activation requires to be happen in this way, and that is what is controlled very critically entire B cell and T cell activation pathways, and that is what we will study in this whole course of immunology. So this is all about the B cell receptor, its activation, its signaling. Let's try to understand one more conceptual term in B cell, and that's called tonic B cell signaling. What do you mean by tonic B cell signaling? Is the activation of B cell without any receptor or independent of any ligand. So the tonic B cell signaling is the activation of B cell without any ligand. So this is ligand independent signaling. This is very low level expression of BCR signaling pathway. So there is very low level BCR signaling that exists, and try to understand it in a different way. So B cells are present in the body, and B cells are required to be there in the circulation for a longer duration of time. If there is no activation, there is no entry of the antigen or say ligand from outside, the B cell has to survive. And to survive B cell, we need to have a low level expression of this receptor, and that is what we call tonic B cell signaling. So it is essential for immature and mature B cell survival. That means they can survive in the body system even without exposure of an antigen. That is what we need to understand. It is primarily activated through PI3, AKT and mTOR pathway. We will try to see this pathway. This is a very critical pathway which is activated upon this tonic B cell signaling. And these are actually very important because without this basic level of activation, the B cell won't survive. So to require B cell to be survive into the system, it requires to activate this cascade, otherwise the B cell will die. So there are the ligand independent say activation where the phosphorylation can happen. So we can see SFK is there. SFK stands for Src family kinases, which are already discussed, and this can phosphorylate immunoreceptor tyrosine based motif, which we have understood, and creating a docking site for recruit the activated SIC, which is again we have discussed the spleen specific tyrosine kinases. So this phosphorylation event of this molecule can activate PI3 kinases, which is again a phosphatidylinositol 3 kinase which catalyze the phosphorylation of PIP2 to PIP3. And if you can recall back in the last class also, we discussed about the PIP3 kinases, which can do a cascade which can lead to activation by doing a diacylglycerol inositol 3 phosphate or inosyl triphosphate, which can recruit the different molecule by channelizing the calcium from the cytoplasmic area. And then AKT is again another molecule which is also called as protein kinases B, and the protein kinase B, and this is activated by PI3 kinase signaling in response to the growth factors. So essentially this protein is getting activated upon even without any ligand activation to the B cell, just to make the B cell survive into the atmosphere. And of course the mTOR is another protein which is called mammalian target of rapamycin, and that's very important because just to survive the host cell for protein synthesis and the cell growth, while this can resist the cellular death by apoptosis. So this cascade is again very important in order to sustain the B cell even if there is no antigen exposure into the system. So this is what you call the tonic B cell signaling, where B cell can survive in the atmosphere without even getting any ligand from outside. So this is all about the B cell signaling, and in the next class we'll try to understand the T cell signaling in details. So the one point to be noticed here is the therapeutic targeting. So when we say that these protein tyrosine kinases can do a cascade here in order to activate the B cell, then there is an auto activation. If there is an autoimmune disease, then probably we can inhibit some of this molecule, and one of the target molecule is SYK inhibitors. So SYK inhibitors such as fostamatinib is tested for the clinical trial in order to make it as an autoimmune disease target. So these are some of the inhibitors which are utilized, such as SYK inhibitor, in order to treat autoimmune disorder. So this is all about the B cell signaling, and we'll try to look into the T cell signaling pathway in our next class. Thank you.